Good day, everyone. This is Dr. Selim, and welcome to our lecture on pharmacotherapy and clinical pharmacy. So, for this session, we will be learning about the complete blood count. So, the complete blood count is a simple way to estimate the physiology of a lot of organ systems that interact to ultimately produce or modify the components of blood. And so the complete blood count details or gives information about the status of the blood at that particular time only when the sample was taken. So, as we have described a while ago, a lot of organ systems interact by way of molecular signals or messengers to ultimately affect the levels of particular cells in the blood. So these organ systems include the bone marrow, and when we see the word myeloid, we are referring to the bone marrow that is active in hematopoiesis, which is the process to make blood cells. So the first organ system that is directly related to the components of the blood would be the bone marrow itself, being the source of all these blood cells. Next would be the renal system, and in particular, this is due to the function of the kidney to produce erythropoietin. Next would be the pulmonary system, and that is of uh, clear reason because oxygenation um, is a direct role of the lungs, and that oxygenation is in partnership with the red blood cell, which holds hemoglobin that holds and releases oxygen into the peripheral cells. And lastly, the gastrointestinal tract as a source of messaging systems and as the way by which the myeloid system is able to procure the precursors for the synthesis of blood. And so, um, as a some sort of bird's eye view, if you have any disorder of the following four major systems, you will have or expect an alteration in the complete blood count. An understanding of the composition of blood as well as the functions and development of each formed element will enable the clinician to derive as much information as possible from the simple complete blood count. So, what does this mean? For you to appreciate the underpinnings of the complete blood count, you need to have a good understanding or knowledge of what composes the blood, and for each of these components, how do they develop and respond to changes inside and outside the human body. So first would be the concept of hematopoiesis. So poiesis means synthesis, and hema means blood. So this would refer to the synthesis of blood. The synthesis of blood occurs in flat bones. For example, the skull, the sternum, the ribs, and the pelvic bones. So these flat bones, in their middle part, they contain bone marrow that grossly is colored red, meaning the softer central portion of the bone, the marrow, is red in coloration. Contrast that to the marrow of the long bones, for example, the femur or the humerus, where the marrow is already yellow to brown in coloration. So when it is already yellow colored, then that means that the bone marrow is now completely filled with fat cells. Here in the flat bones, example, what I have told you a while ago, the marrow is red colored, meaning there are active stem cells that continue development to become the mature cells found in the blood. So, you can think of it this way, that the red bone marrow, the myeloid system, and environment is actually like the kindergarten of the blood cells. And once these blood cells complete their maturation, they now exit the bone marrow to then occupy the blood. Stem cells progressively become more specialized in the presence of unique hormones or signals. So initially, the stem cells do not know what they need to become. But, for example, there is a stimulus, and then there is a need for the body to adapt to that stimulus. There will be hormones that are released, and those hormones will tell the stem cell to specialize into this particular line. For example, the presence of a bacterial infection, for example, in the lungs, will release interleukins. And these interleukins, as they float in the blood, they then go into the bone marrow and tell the stem cells of the bone marrow to become white blood cells so that response to the infection is now possible. So this picture shows you how the different cells that you see in the complete blood count develop from their more immature forms into their mature forms. So just follow the cursor and bear with me. Here at the bottom, you will see the red blood cell or erythrocyte, the basophil, the neutrophil, the eosinophil, the monocyte, the B and the T lymphocyte, and the platelets. These are the formed elements that you will see in the blood. Now, if you will trace it backwards, as you go more superiorly in this image, you will actually end up with the hematopoietic stem cell here. And so, if you follow the different lines, they all start at the hematopoietic stem cell. This is just one undifferentiated cell, the same cell for all of these different cells at the bottom. So that goes back to the point that we have discussed a while ago. What tells the hematopoietic stem cell to become, for example, a platelet, or to become a monocyte, to become a neutrophil, to become an erythrocyte? Those would be the signals that are produced in response to a change or stimulus. Okay, so first you have here the different portions of the diagram that are color coded. First would be this light pink portion right here. This means that all the cells that are included in this light pink area are still within the bone marrow. And then there are cells that are released from the bone marrow, and this is coded by light blue. And then from this point, they now become intravascular forms, meaning these are the actual cells that you will see in the complete blood count. And more so, some of these cells can actually exit the blood, exit the space inside the blood vessel to actually go into the peripheral tissues. So for example, in our case in point a while ago, we talked about pneumonia. And in pneumonia, there is for example, a bacterial pathogen that has invaded the lung. The response of the lung would be to release interleukins that goes into the blood from the lung. Then those interleukins would then go into the bone marrow and tell the stem cells of the bone marrow to increase the production of this line of cells. So the hematopoietic stem cell pool or reservoir then diverts into becoming these cells right here. And once released from the bone marrow, there will be again active production of these cells. And then from the bone marrow, these cells go into the blood. And in particular, the neutrophil goes into the blood, exits the blood, and goes into the substance of the lung, in our case in point, in pneumonia. The neutrophil, which is in the blood, now goes into the lung. And then in the lung, it phagocytoses the pathogen and eventually forms part of the phlegm. Okay? And so, whenever the mucus that is produced in the airways of the lung goes up and up and up into your mouth, when you spit the phlegm and it's colored yellow or greenish, that means there are a lot of neutrophils in the phlegm. But the question again is, where do these neutrophils come from? From the bone marrow. Okay? So, here you have the different stages of the development of the red blood cell. What do you notice in this picture? The earlier forms of the red blood cell still have nuclei. Okay? But eventually there will be nuclear extrusion. The red blood cell throws away its nucleus so that it has space inside, so that that space, now freely available, becomes a space occupied by hemoglobin, so that the mature red blood cell is actually fully filled with hemoglobin. The reticulocyte is an immature form of the red blood cell. It is already released from the bone marrow, but there are still fragments of the endoplasmic reticulum of these cells. And so the endoplasmic reticulum produces this appearance. But eventually, it matures to become this biconcave erythrocyte. Here, you have the line for the development of the granulocytes. We call the granulocytes as such because these are white blood cells that contain small dots or granules inside. Okay, so a basophil is a granulocyte, an eosinophil is a granulocyte, and a neutrophil is a granulocyte. The functions of these we will have a discussion of later. And then you have monocytes. What's special here is that the monocyte, if you will compare it to the granulocyte, these are also white blood cells. But the nucleus of the granulocytes often are multiply lobed. For example, here in the neutrophil, you have 1, 2, 3, 4, 4 lobes. But the monocyte has one big nucleus. Thus, you call it a mononuclear cell or monocyte. The monocyte that is in the blood goes out of the blood and into the tissue to become a mononuclear phagocyte or macrophage. So the macrophage is actually the extravascular form of the monocyte. And then you have here the lymphocyte line. What's special about the lymphocyte line is that they come from a different um, line of cells. Okay? From the hematopoietic stem cell, there will be a committed lymphoblast, which will eventually become the B lymphocyte and the T lymphocyte. What differentiates the two? These two cells actually come from the same progenitor, which is your prolymphocyte, but their further stages of development occurs in different tissues. For example, for B lymphocytes to become mature, they have to go from the bone marrow and find their way into the spleen or the lymph node. And in the spleen or the lymph node, that is where they will mature. But the T lymphocyte, from being inside the bone marrow, goes out of the bone marrow into the thymus, which is an organ or gland found in the chest. And there in the thymus, it becomes the T lymphocyte. So, what differentiates B and T? Their sites of maturation or development. That is why B stands for bursal and T stands for thymic lymphocyte. And lastly, the platelets. As you will see, the cells that um, become the precursor of the platelets actually are very large cells compared to here in the left. And so these are very large cells with very large nuclei. And therefore we call them mega, meaning large, megakaryocytes. The megakaryocytes actually line the spicules of bone in the bone marrow. And as they go into the spicules, they fragment to become the platelets that fill the blood. So, in summary, in this picture, we have discussed the formed elements that we see in the blood. What were they when they were still in the bone marrow? And what will they become once they exit the blood? What is the composition of blood? First, you have to understand that blood is a tissue, meaning it is composed of several cells. Blood is not an organ, it is a tissue. So, the formed elements, the cells, white blood cells, and corpuscles, for example, the red blood cells and the platelets, are suspended in plasma. So they are floating in the plasma. The formed elements may increase or decrease in quantity based on the state of the patient. For example, when the patient loses a lot of blood, the red blood cells on the first day of acute blood loss, example, 2 liters of blood are lost. The red blood cells may still be normal, but 2 days later, the quantity of red blood cells in the blood goes down. Another example, an increase in the number of white blood cells in the blood happen as a result of severe infection. And that is a way of increasing the cells that actually fight off the infection. Or what may happen to the formed elements is that the nature and the characteristics may be altered sufficiently to produce dysfunction. For example, when a patient is exposed to a diet that is poor in iron, what can happen is there will be insufficient iron to produce hemoglobin. And so the amount of hemoglobin inside the red blood cells will decrease. And that will change the color of the red blood cells from being deep red to pink. Okay? They become paler. And because they have little hemoglobin inside, the ability to carry blood also, the ability to carry oxygen by the blood also decreases. Plasma characteristics, on the other hand, is directly the effect of water balance and synthesis of factors and the mechanisms that control them. For example, let us go back to the basic concept of hematocrit. Hematocrit is the amount or volume of blood percentage occupied by red blood cells. So, for example, the normal hematocrit is 35 to 45%, meaning if you have 100 ml of blood, 45 ml of that would be occupied by the red blood cells. Now, what do you think will happen to the hematocrit if the patient suffers from dehydration? The amount of water in the body decreases, and so the volume of plasma decreases. So, from an original percentage of 45%, there will be an increase, a relative increase in hematocrit because the volume of plasma which suspends the red blood cell decreases. So this picture right here is how the kidney influences the development of red blood cells by secreting the hormone erythropoietin. So, for example, there are these factors: disease of the lung, poor blood flow, low amount of hemoglobin, example in iron deficiency anemia, anemia itself, and low blood volume, for example in severe chronic dehydration, or if you have acute blood loss. What will happen is that there will be decrease in oxygenation of the tissues. Okay? If there is decreased tissue oxygenation, the tissue of the kidney senses that there is now little oxygen. And so in response, it releases the hormone erythropoietin that goes into the bone marrow and tells the stem cells to commit to become red blood cells. And so the hematopoietic stem cells become red blood cells, and that increases the tissue oxygenation. And so this is your negative feedback mechanism for oxygenation. Red blood cells have specialized to carry oxygen and carbon dioxide for transport between the lung and the peripheral cells. So how did they specialize to carry more oxygen? They have extruded, they have thrown away their own organelles, including the nucleus, to make space for hemoglobin so that all of the volume of the red blood cell is filled by hemoglobin. Next, the cell membrane of red blood cells have proteins that facilitate the entry and exit of bicarbonate and carbon dioxide. So, for example, the carbon dioxide that is in the peripheral cells is converted to carbonic acid and then to bicarbonate inside or on the surface of the red blood cell. The bicarbonate goes into the inside of the red blood cell, and as the red blood cell now goes back into the lung, it then releases the bicarbonate, which then becomes carbon dioxide again for exhalation. Next, important for the health of the red blood cells, for the characteristics of the red blood cell, would be adequate levels of iron. Why? Iron is the central atom in the heme molecule. So the heme combines with globin to become hemoglobin. And so if you have low levels of iron, you won't be able to form heme. If you are not able to form heme, you are not able to form hemoglobin. On the other hand, folic acid and the other B vitamin, cyanocobalamin or cobalamin, they are essential for transferring methyl. So, for one carbon transfers, folic acid and cyanocobalamin are important. So, what then happens if you have low levels of folic acid? The DNA, which requires four nucleotides, adenine, guanine, cytosine, and thymine, will not be able to efficiently form, the cell will not be able to efficiently form the four nucleotides. Why? Because for you to form cytosine, you need to have adequate levels of thymine. You have to methylate the thymine to form cytosine. If you do not have folic acid, there will be no interconversion of thymine and cytosine. So that for the four nucleotides, you will lack one of the four. Okay? Because there is no methyl transfer, because there is no folic acid. And so what will happen to the nucleus of the developing red blood cell? It will become abnormal. It will not mature. It will not be able to decrease the size of its nucleus. Why? There is no methyl transfer, there is no DNA. This picture shows again the development of the red blood cells. And so we should end up with red blood cells that look like this. You have biconcave discs, okay, and that will increase the surface area of the red blood cell. For example, you have low levels of iron. What will happen? Hemoglobin synthesis will be inadequate. And so there will be microcytic cells, cells that are micro or small. Why are they small? Because they are not filled with hemoglobin. They do not enlarge because there is no hemoglobin that fills them. Okay? We have hypochromic cells. Why are they hypochromic? They have low coloration because there is little hemoglobin that colors the cell. So, if you have iron deficiency anemia, you will have small cells, microcytic, and pale cells, hypochromic, microcytic hypochromic anemia. Then next, as you will observe, your mature red blood cells should look like this. But if you have megaloblastic anemia, as what will happen in deficiency of folic acid and cobalamin, you will have cells, red blood cells, that have large nuclei, that are large and irregular in shape. Some will be filled with hemoglobin, some are not. Why? Because the nucleus failed to develop appropriately. And so, if the cell should normally shrink during red blood cell development, as you will see here, if you will impair the development of the nucleus, you will have large cells that have failed to shrink. Next, neutrophils are white blood cells, granulocytes, that are the first responders to infection and tissue injury. They develop when the hematopoietic stem cell is exposed to granulocyte macrophage colony stimulating factor, GM-CSF. Eosinophils, on the other hand, are also granulocytes that increase in number during allergy and parasitic infections. So, for example, if you have infection of the colon with amoeba, you will have an increase in eosinophil. If you have the presence of worms in the colon, you will have an increase in the numbers of eosinophils. If you have an acute asthma attack, you will have an increase in the number of eosinophils. What will then tell the hematopoietic stem cell to become an eosinophil? The presence of interleukin 5. Interleukin 5 is increased whenever the host or the organism is exposed to a parasite. Basophils, on the other hand, increase during massive histamine release disorders. So for example, if you have an anaphylactic shock, or if you have anaphylaxis, that's a severe allergic reaction. Remember that the terminal mediator of anaphylaxis is histamine. So if you have severe anaphylaxis, you will have an increase in the basophils. This table will show the function of the different formed elements. Platelets are important for hemostasis to prevent bleeding. Platelets promote blood clotting, and whenever the inner lining of the blood vessel is injured, the platelet plugs the damage. Neutrophils will eat bacteria, and neutrophils will increase in number in acute bacterial infection. Eosinophils phagocytose or eat up antigen antibody complexes. And so, if you have a lot of these complexes, for example, an allergen being exposed and then to the patient, an allergen being exposed to the patient, and an immunoglobulin E complexing with the allergen, you will have an increase in eosinophils. Basophils are involved in anticoagulation because they have a lot of plasmin inside the cell, and basophils also increase vascular permeability through the presence of histamine inside the basophil. When basophils leave the blood and they go into the tissues, they become mast cells. Monocytes are motile, and once they are already in the tissues, they become macrophages, which are very efficient in clearing bacteria and debris. Lymphocytes are important for adaptive immunity. These are more powerful immune responses. So here is a picture, the blood picture of a patient with iron deficiency anemia. Remember that in iron deficiency anemia, there will be decreased hemoglobin synthesis. So you will see that here. For the normal individual, the hemoglobin is 14, and for this patient is only 10.8 or 11. What will happen to the hematocrit? Hematocrit is the volume occupied by red blood cells. If you have iron deficiency anemia, you will also have less red blood cells or smaller red blood cells. Therefore, they will occupy lesser volume in the blood. From normal of 41, here you have 33. In the picture that we have discussed a while ago, what is the usual description of the red blood cells? They are microcytic, meaning they are small red blood cells. So, if the normal size of each red blood cell is 86 femtoliters FL, your patient with iron deficiency anemia will have smaller red blood cells, only 75. MCV is your mean corpuscular volume. So it will tell you the size of each red blood cell. So from normal of 86, you have a smaller red blood cell of 75. MCHC and MCH will tell you the amount of hemoglobin present in each red blood cell. And so if the normal person has his CBC, the amount of hemoglobin inside would be 33.6 grams per deciliter. If you have iron deficiency anemia and you cannot make any hemoglobin, the amount of hemoglobin per red blood cell decreases to, for example, in this picture, 30. And all the rest um, will be within normal. Next, what will happen if you have low levels of folic acid? The development of DNA and the nucleus in the red blood cell will become abnormal. And so you will have large red blood cells. So this would be serial readings in a patient. So this is the same patient. And in this report, they are following the patient through weeks of giving folic acid. So initially, the hematocrit of the patient, the amount, um, the volume of blood occupied by red blood cells is 17.9. After giving the patient folic acid, the number of red blood cells now start to increase and they now become more mature red blood cells. So from 17.9% after giving folic acid, it now becomes 38%. So this is now normal. The mean corpuscular volume, telling you of the size of the red blood cell. Before giving folic acid, it is 93.6. So that's very big. The red blood cells are very big. Now, if you start giving the patient folic acid, it decreases to 77. The cells become smaller because they are now more developed. Okay. So for megaloblastic anemia, we are concerned with the mean corpuscular volume. The cells are bigger than usual. Okay? Here you have pictures of platelets. What I would like to emphasize is that inside the platelets, you have granules. Okay? So example, here and here. These granules or vesicles contain mediators. They contain substances. So for example, one of the substances found inside the platelet would be serotonin. And once the platelet releases the serotonin, the blood vessel will start to vasoconstrict so that bleeding stops. Another molecule found inside platelets would be histamine. Another molecule would be thrombin and fibrin. So again, what would be the function of fibrin and thrombin? These are substances that help cement the clot. So platelets release the clotting factors so that the clot becomes more stable. And this is what a clump of platelets would look like when viewed under the microscope. So you can think of platelets as having a role in hemostasis and even in inflammation. And so whenever the platelets increase in number, that means there is active inflammation and there is active bleeding that needs to be stopped. But the other way can also be found. If you have a patient who has a lot of bleeding for a very long time, what can happen is the platelets can decrease in number. Why? Because they are now being consumed to stop the bleeding. And so the amount of platelets that you draw from the blood actually are decreased because they are now sticking to the blood vessel wall that is injured.